everyone, I am Jedaidah, your research instructor, and welcome to the end of week 3 of our research course. We are almost here and you have done so much so far. So next week is our last week of our research course, and you are going to finally communicate your research work, your scientific method that you've been working on for the past 6 weeks, including the 2 pre-class weeks. You will communicate that through a research paper and a poster presentation. And so all your hard work will come together in that, like, those two communication formats. So all the work you've done in filling out your eLab notebook every week, you always update your eLab notebooks talking about the title of the experiment, why you did it, how you did it, the methods, what results you got. And then you also every week you include photos of your results in the photo uploads. But all these things are going to come and be communicated. You will write all these things in your research paper and your poster presentation. So I'm very excited to see that, and it's to really learn, because we're all learning what's actually happening in your own city and state when it comes to the microbiology, the plant microbe interactions, and bioinformatics tools that we're going to use. Okay, so in this video I'm going to demonstrate how to isolate bacteria from roots. So we did this yesterday in class, and for every experiment that we do in class, we do an experiment in one location or one pot, and then in your own time you have to repeat that for the rest of your pots. So by the end of the week, before Sunday, by Sunday, you then update your eLab notebook, and you also take photos and update them in your photo uploads. Okay, so this video is going to become a shorter version of what we did yesterday, because you have an idea of what that is, but just so that you can refer to this as you continue doing the experiment for the other locations. And always remember that you might do experiments for each location on different days. That's completely fine, as long as you indicate the date, because that's something that is really important in case your results show some discrepancy between location. It could be because there's been a gap in when you analyzed. Okay, and that's completely fine as long as you indicate. That's why you have your eLab notebook, and then you have your photo uploads to record your work. And remember, as we said, in science, it's very important for us to record every single thing that happens. Okay, so how do we isolate bacteria from our roots? What do you need? Okay, so first of all, you're going to need agar plates that have media. Media is essentially a jelly that has different chemicals that are perfect nutrients for bacteria to grow, or fungi, in general microorganisms. And so there are different types of media, which we'll talk about in the next class. So you have this particular one. Remember, you kept this in the refrigerator, so you need to take that out. So you have that, and then you have your envelope in a lab kit that says microcentrifuge tubes and plastic pestles. So this is essentially, so these are the plastic pestles. They are individually wrapped, so they're kept sterile. Okay, you will need one for each location, each pot. And then you have the microcentrifuge tubes. You should have 5 or 6, 5. And so to start with, you just need 3. Okay, 3. One of them will be the one where we will finally do our isolation. We'll crush our root sample, our root segment, crush them. Once that is done, you can just trash it. And the other 2 tubes, you can actually use them. One of them will fill them with just clean water. Then the other one, we will dilute a little bit of bleach and add a lot of water, and then we mix it up. So the first one is going to be our bleach diluted solution, and the purpose of that is to sterilize our root segment. And the second one is going to have water, clean water, and that is to then rinse off that bleach solution that has been used to like sterilize our root segment. And the third one is where you finally put your sterilized and rinsed off root segment, and we're now going to crush or homogenize our root segment using the plastic pestle. And why do we like crush that root segment? Okay, so first of all, why do we have to sterilize? We have to sterilize because we want to get rid of any other microorganisms that might be around or outside our little root segment that we're going to cut off. Because remember that this is coming from the soil. The soil contains many different microorganisms, good, bad, harmless, but we are only we are only concerned about one particular type, which we know enters the plant roots. Okay, and then we rinse off the bleach, because we don't want that bleach to eventually get to our target bacteria and then kill them. And then the third one, we crush or break open our root segments. Why is that? Because we are looking for bacteria that is like inside these roots. Okay, so whether you have root nodules or not, we will still do this. If you have no root nodules, then just cut off a segment of the root, because remember that in the signal exchange, when nitrogen fixing bacteria and legumes actually interact, first they communicate. There's this like molecular or like biological communication where the plant sends signals to the bacteria. The plant sends signals into the soil to attract a particular bacteria, and then the bacteria then sends signals back. Then when that communication is successful, then the bacteria move towards the plant roots, enter the roots, then eventually root nodules are formed, and then nitrogen is provided in the root nodules. So it's possible that if you don't have any roots, maybe it's that initial stage, like the communication has happened, some bacteria in there, but they haven't formed nodules. Or there could be other kind of bacteria which would be in your roots that don't induce root nodules. So we are all isolating bacteria from our roots. All right. Okay, so have those three. We have that, and then you need label. So today we'll actually label all three, so we actually like keep track of what these are. And then you will need a scalpel. So this, you have one that comes sealed. I unsealed mine yesterday when we started doing an experiment, and so if yours is still sealed, take it out of the package, and then you have to like take off the plastic clip so that that blade is exposed. Okay, and once you use it for the first time, the next time you use it, please use your wipes to wipe the whole scalpel. Okay, and then make sure you get your gloves. So I'm gonna start this, and I have my gloves. Oh, and then the last thing is you need household bleach. So like I said yesterday, household bleach, for most of us, the percentage of the active ingredient, sodium hypochlorite, was like 6%, 7%, 8%. And so we have to dilute it where we have very little bleach and a lot of water. You don't want to be like super concentrated. We need to be like just like a low concentration of the bleach. And yeah, so I have mine. So the easiest thing to do is, because you only need a little to add in one of these tubes, get your household bleach, put like on a lid or like a small Tupperware, then it's actually easy to like pour it in one of these tubes. Okay, so let's get started and let's do this. we need to get our plants, and the first thing we have to do is do a second shoot phenotype. You already know how to do this. And then do a root phenotype using a microscope, and then on the same root cut off a segment that has nodules, okay, or your nodules, or as many as you can get that will fit into the tube. Just kind of like near the bottom. If you don't have any root nodules, just get a big enough segment, and then you add it in your tube. Okay. So first things first, let's label our tubes. All right, so I have label around. So one thing you have to remember is that when you look at these microcentrifuge tubes, they actually have some markings on them, and some of them have numbers. So you have like the first unit mark at the bottom, then the second one has a number, that's 0.5, and then there's the next one, no number, then 1.0, 1.52. Those are the units for mils, or milliliters. And so I will refer to these when it comes to like how much do you add. So is it going to be below the 0.5 mark? It's going to be on the 1.0 mark, or the 2.0 mark, or 1.5. So remember that. So even when you put your label, put your label like away from the marking so that you can actually see. And what do you put on the label? On the first label, please put the location that you're doing. So for me, it's going to be walkway. So you can use a Sharpie or a pen. So walkway. And then on the other one, you can just put water. Okay. And in the third one, you put bleach. Okay, so I have my 3. Okay. And then one thing that I forgot is that we need clean water. Okay, clean water that we're going to work to make our dilutions, and then later on put in the pot to help soften the soil so we can actually dig up our plant, and then rinse off the soil particles. All right, so let's fill our 3 tubes with the necessary solutions, the ones I need. So I'm just going to sterilize my hands again. Okay, so one is for clean water. So I'm just going to get some water from my tap, scoop it out, and try and get water up to the 2.0 mark or 1.5 mark. So any of those will be fine. Next is the bleach tube. So like we said yesterday, you only want very little bleach and a lot more water. It has to be diluted, and so a drop or 2 drops of water should be way below the 0.5 mark. So you know how like there's this like little like V shape? You want your drop to just be at the bottom, and it should not even like reach that mark. If it reaches that mark and you can't take it out, then just make sure you really fill this up to like almost the brim. Okay, so you want to have very little, like a drop of bleach, and it's easier if you put like on like I have mine like on a lid, because the container is really big, and when you pour it, you might end up like pouring too much. So I'm just going to put very little of it. All right, and then my next part is I have to add water. So it's actually easier if we add water from the other tube, and then we can actually get some more later. So I am going to put this to the actually in this case, because mine is like 8%, I'll put this to the 1.5 mark. Okay, so remember that everything that you do, however number of drops you add, the volume that you use. Is it like 1.0 ml or 1.5, depending on like where the level was at. You have to indicate that in your eLab notebook. So if you're on the high concentration, add more water, and if you're more on the low concentration, you obviously add less water. Okay, just by inverting. So I have my bleach tube filled, and then I can go back and just like fill this with the water. Okay. All right. Okay. Then your third one, which has the location, you don't put anything in there, because eventually you're going to put your root sample. So I will have that over here. Okay, great. So now first thing is I want to collect my plant, so I can first phenotype the shoot and then phenotype the roots, and then from there you then use the same roots, and then we carry out our bacteria isolation. All right, so